In order to stain a bacterium, we must first prepare the organism on a slide. First, a bacterial smear is made of the organism. This is simply made by placing a drop of water on a microscope slide . Next, by using proper aseptic technique, we will emulsify a culture of the organism in the droplet. Please remember to flame your loop after obtaining your specimen. Next, in order to prevent the organism from being washed off from the slide during staining, the slide is allowed to either air dry, or to speed up the process, we can pass the slide through a flame. This particular part of the procedure is referred to as heat fixing the specimen. Once the organism has fully dried, you should see a haziness on the slide. To heat fix, grab your trusty clothespin and use it to hold the slide like so. Pass the slide through the coolest part of the flame toward the bottom a few times and hold it to the side. Repeat this process until the water droplet has evaporated. You can tip the slide to thin out the droplet to aid in the drying process. Due to the nature of how a drop of water dries, the droplet will dry from the outside in, but while it does this, the organism will be drawn to the center of the fixed area. Because of this, the bacteria will be thicker and dense in the center. Therefore, it will be necessary for you to focus your attention on the outer periphery of the fixed smear in order to be able to view individual bacterial cells. It is not necessary to place a cover slip on a fixed bacterial smear since the organism will not come off the slide. Oil can simply be placed directly on the specimen and viewed using the 100x objective. You will begin to see your water droplet dry and bacteria will be fixed to the slide. Care should be taken not to overheat your slide during this process. Be sure to properly label your slide. Labeling with a Sharpie marker while doing some staining procedures may wash the marker off the slide, especially when you're using ethanol or decolorizer or anything like that. So in order to prevent this from happening, please use a pencil to label your slides. As you can see, the water droplet is getting smaller. You do not want to rush this part of the process, and that's how you conduct a bacterial smear.